Thank you very much. Thank you very much for the invitation, and thanks to Norberto for organizing this course. It's very nice to be here. My topic today is to review with you some of the aspects of fibrogenesis in non-alcoholic fatty liver disease. Let's see if I can work this out. Oh. Okay, these are my disclosures. So we have learned already today that the relevance of fatty liver in the context of hepatology and the very worrying epidemiology. So we as hepatologists all know that the fatty liver disease is actually a spectrum that encompasses simple fatty liver through steatohepatitis, fibrosis, and cirrhosis in a fraction of patients. So it's very important to understand the mechanisms that lead from steatohepatitis to fibrosis and cirrhosis, because these are the patients who are more at risk to develop complications of liver disease. We also know from epidemiological studies that patients with fibrosis are the ones who are more at risk of mortality, not only of liver-related mortality, as it is understandable, but also of non-liver-related mortalities. This is a classical study from Sweden showing that patients who have NASH have more risk to die in a long follow-up. And an eventual follow-up that was published a few years ago in Hepatology shows that fibrosis is the only predictor of death in these patients. And this is mortality due to all causes, not just liver diseases, but also cardiovascular diseases. And similar findings were published early last year by the group of Rohit Lumba, who showed pretty much a similar thing, that increasing the stage of fibrosis poses a higher risk of mortality due to all causes in patients with NASH. So fibrosis is probably the single most important component of the spectrum of non-alcoholic fatty liver disease that we should be aware of. So what is fibrosis? Fibrosis is actually a process of wound healing. The liver is wounded, in this case is wounded by an injury due to the lipids, as we have heard by the previous talks today, by lipotoxicity. And the lipotoxicity injures the cells. This leads to inflammation, regeneration, and repair under the form of deposition of fibrillar matrix. All these processes are not coordinated and eventually lead to fibrosis and cirrhosis. The timing of the injury is very important, because we all know that if the patient has an acute hepatitis that results, for example, acute hepatitis A, there is no fibrosis that is residual in these patients. But if we have a repetitive and continuous injury, the amount of fibrosis progressively increases and the patient ultimately goes to cirrhosis. We have already heard about the relevance of some of the cells that are within the liver, especially the stellate cells. These cells have a quiescent phenotype in the normal liver. They store vitamin A, but in response to all sorts of injury, including fat, they undergo a process of transdifferentiation into fibrogenic cells that produce extracellular matrix and cytokines that coordinate the fibrogenic responses. So the biology of stellate cells has helped us a lot in understanding the processes that lead to fibrogenesis. If we go more specifically into the problem of steatohepatitis, basically we have hepatocytes that undergo a metabolic injury, that is basically the accumulation of triglycerides and lipids. We have heard this and the mechanisms in the previous talks. Some of these patients, however, associate a lipotoxic injury to the metabolic injury that is simply the accumulation of fat, and this leads to the activation of fibrogenic pathways, the wound healing, and the NASH fibrosis. So some of the aspects that have been studied in the past years have led to more understanding of this process. One of the things is genetics. We have heard of genetics. We know that PNPLA3 is very prevalent. You know, here genetics is very easy to understand. If you breed these two subjects, you get a mixed phenotype here. So this is a way to understand the genetics that is pretty easily. So some of the genetic factors have been unraveled lately, including PNPLA3, TM6SF2, MBOT7, and MERTK. And what I'm going to show you is how, from the genotypes, we are accumulating evidence that makes us understand the mechanisms from the genetic factors to the actual fibrosis. For example, TM6SF4, this is work mostly done in the United Kingdom by Quentin Anstey, is very interesting because depending on the genotype, it may confer a higher risk of developing NASH and fibrosis as opposed to increased VLDL excretion leading to higher susceptibility to atherosclerosis and cardiac events. Another gene that has been investigated is PNPLA3, and this is a study showing that patients carrying the at-risk allele have a higher risk of having NASH and having fibrosis. So recently, in collaboration with the group of Michael Trauner in Austria, we have investigated the role of the mutation of PNPLA3 in hepatic stellate cells. We found that the PNPLA3 is expressed in hepatic stellate cells, and more importantly, when these cells activated, as you can see following the alpha SMA expression, they also increase the expression of PNPLA3. And this is associated with the classical phenotypes of the activated stellate cells, including increased collagen and decreased PPAR gamma. The interesting thing is that if we isolate cells, stellate cells, from subjects that have the mutation of PNPLA3, we observe that the pro-inflammatory phenotype is more evident. You can see a number of cytokines, including interleukin-8, TGF-beta, CCL5, are expressed at a higher level, and the supernatants from these cells have a greater ability to recruit macrophages to the sites of injury. And this is just a cartoon, a summary cartoon that you will find in the paper that shows that through a number of signaling pathways, including Jun kinase and downregulation of PPAR gamma, we believe that PNPLA3 may contribute to the induction of the profibrogenic phenotype of stellate cells. Similar is another genetic factor that is MERTK. Here, the MERTK allele at risk is associated with a higher expression within the liver. The patients who carry the G allele have more MERTK. MERTK is a tyrosine kinase. It's actually a transmembrane protein, and here also have found that activation of MERTK regulates the biology of human fibrogenic cells, because if you expose these cells to Gas6, which is the ligand, one of the ligands of MERTK, we induce a higher migration of the cells and pro-collagen expression. This pro-collagen may be inhibited by a receptor antagonist of MERTK, this UNC569, and this also leads to a lower viability of the cells. So MERTK is important for inducing collagen expression, but it's also important to induce viability of the cells. We are currently also exploring the role of MERTK in macrophages. You can see that it's pretty abundant in macrophages in patients with NASH, and this is an ongoing story that we are currently investigating in the lab. The important thing is that the MERTK belongs to this family. It's called the TEM family of tyrosine kinase receptors that includes also AXL, and both receptors have been recently implicated in the pathogenesis of NASH. This is work coming from Spain, from Barcelona, showing that also the receptor AXL is relevant for the pathogenesis of fibrosis in NASH, because animals treated with the antagonist of AXL have reduced in vivo fibrosis and reduced hydroxyproline, that is a marker of fibrogenesis in vivo. One important correlate of this data is that we should be aware that there is a familiar and genetic aggregation of NASH and of the risk of fibrosis. This is a recent paper from the group of Rohit Lumba showing that there is a higher risk of fibrosis in relatives of patients with NASH cirrhosis. We as hepatologists must be aware about this, because we have to collect family history in all patients that come to our clinics, ask them whether their, especially the first-degree relatives, were diagnosed with fibrosis or cirrhosis, because this may pose a higher risk of progression. So in the next minutes, I will also review some of the other mechanisms that lead to fibrogenesis in the context of steatohepatitis. And here I have listed a number of different factors, including lipotoxicity. We have already heard a lot about lipotoxicity. I will not dwell on this topic too long. The important thing that the other speakers have already highlighted is there are sort of like good fats and bad fats, including ceramides, free fatty acids, free cholesterol, and these are more likely to induce lipotoxicity and be associated with NASH. There are several mechanisms by which the lipids may induce damage to the hepatocytes through direct interaction with receptors, through mitochondria, through direct activation of intercellular signaling pathways, and one of the mechanisms that have been highlighted is the activation of the ER stress. ER stress is a complex response of the cell. It's actually a defensive response that is called as the unfolded protein response, but when this is not sufficient to maintain the viability of the cells, there is an activation of a pathway that is called ER stress that is associated with insulin resistance, cell toxicity, and inflammation. And recently, a paper showing that a molecule interference with the IRE1, that is a critical component of the endoplasmic reticulum stress, is relevant for the pathogenesis of NASH. Here, it has been tested a BAX inhibitor 1, that is a protein that associates with IRE1, and this molecule binding IRE1 protects from NASH. And we can see this especially looking at the expression of pro-inflammatory cytokines and the effects on cellular apoptosis. Another way by which lipotoxicity may be active is through cell-cell communication via microvesicles. This is a study from a group of our collaborators in Torino, in Italy, showing that microvesicles from injured hepatocytes, and these hepatocytes were injured by the way of a lipotoxic stimulus, including palmitic acid, resulted into generation of microvesicles that activated some pro-inflammatory pathways, including NLRP3 inflammasomes. We will say something more about inflammasomes in a minute. In fact, inflammatory balance is another factor that is very relevant for the pathogenesis of NASH. Here we have to understand that the stellate cells, which are the ultimate producers of collagen and extracellular matrix, are actually in a milieu where a number of soluble mediators are released by macrophages, sinusoidal endothelial cells, and hepatocytes themselves have the ability to modify the biology of stellate cells. And macrophages are very important. They are relevant in the process of initiation of liver injury when cytokines released by the monocytes and activated Kupffer cells. These are activated monocytes that have high expression of LY6, release several cytokines, including MCP1, CCL2, that leads to the transition of stellate cells to myofibroblasts. But we have to point out that macrophages with another phenotype, the so-called LY6C low monocytes, release a number of matrix metalloproteinases, which are important in the process of fibrosis resolution. So it's not just whether macrophages are good guys or big guys, it really depends on the phenotype of these macrophages. And here also, these are very tightly regulated. This is a study that has just come out in hepatology, showing that hypoxia, that it's a factor that occurs in liver injury and especially in NASH, leads to activation of hypoxia-inducible factor 2 alpha, which in turn leads to the upregulation of this histidine-rich glycoprotein, which is relevant to modified macrophage polarization toward a more pro-inflammatory M1 phenotype. Another way by which the recruitment of monocytes is regulated is through chemokines, especially CCL2 or MCP1 and CCL5 of rentis, and this is already into translation because we have a molecule that is called cenicriviroc that is in phase 3 study for NASH. I think we will hear more today when we will talk about the therapy of NASH. But this is also very effective in animal models, even in the so-called therapeutic models. This is a model of steatohepatitis carried out for 4 weeks. After 4 weeks, cenicriviroc is administered to these mice, and this is already sufficient to reduce inflammation and also to reduce fibrosis. And hopefully this will occur also in humans. We will know hopefully soon. The inflammasomes have received considerable attention. These are molecules that sense a number of exogenous stimuli, including bacterial-derived products and damage-derived products from damaged cells. Here are a number of different inflammasomes. The one that has been more studied in more detail is the NLRP3 inflammasome. This inflammasome pathway is activated not just in animal models, but also in NASH. This is a study from the group of John Gisabo. But the interesting thing is that inflammasomes are relevant not just in the liver, but also in other tissues, including the gut. And they might be regulators of the liver-gut homeostasis. It was shown a few years ago that the mice that have that are defective in the inflammasome pathway have altered microbiota, and this leads to greater activation in the liver of toll-like receptors like TLR4 and TLR9, leading to pro-inflammatory signals and NASH. And this is something that occurs in the intestine, leading to a changed microbiota, and was also confirmed by another group in Italy, the group of Svegliati Baroni, showing again that the lack of NLRP3 causes dysbiosis and leads to inflammation in the liver and in the adipose tissue. So apparently, if we look at the inflammasome in the intestine, its reduced activity might be detrimental. But what happens in the liver? In the liver, we have a different view. If there is constitutive expression and activation of one of the components of the inflammasomes in the liver, we actually observe an increased fibrosis. And on the other side, if we, and this is with a knock-in model, that is a model where the mice are genetically engineered to have an activated inflammasome. Inflammasomes are regulated by different factors, including fatty acids, are maybe mediators of lipotoxicity, and interestingly may be regulated by not only by drugs, but also by different herbal compounds, including silibin. Silibin seems to modulate inflammasome activation through activation of sirtuins, and especially sirtuin 2, and also other but also other herbal compounds might be important. This is like, this is an editorial that came out in Cell several years ago showing that we should actually look for the mechanisms that lead from the traditional medicine to actual drugs. And one example is berberine. Berberine is very much used in the United States, and a couple of years ago we found that the berberine modulates inflammasome activation associated with steatohepatitis. This is the MCD model where you can see that expression of interleukin-1 beta and other components of the inflammasome system are decreased by berberine. And interestingly, this occurs through a membrane receptor that is a purinergic receptor called P2X7, which binds ATP. ATP is released by dying cells, and berberine appears, at least in part, to work on this receptor. And this shows a sort of translation from traditional medicine to what we see in, from what we can see in a more molecular fashion. Another possibility to interfere with the inflammasome pathway is to interfere directly with interleukin-1 beta, and there are studies ongoing, for example, with the IL-1 receptor antagonist, although this may pose some problems, especially in terms of risk of infection. Yeah, I will skip a couple of slides. Some other mechanism, can I have a couple of more minutes? Yeah. Some other mechanisms that have been implicated are, of course, the nuclear receptors. Nuclear receptors have been implicated in fibrosis for many years, especially PPAR gamma, and now other receptors, especially FXR. FXR is under considerable investigation in NASH because it has effects not just on fibrosis, but also on inflammation and some of the metabolic pathways associated with NAFLD, including the FGF21 axis and the liver-gut axis, and it's a very promising therapy. Here also we have a phase 3 study ongoing. Ductal reaction is a way by which the lipotoxicity induces the activation of progenitor cells that appear as ductal reaction, and here it was shown that along the stage of fibrosis you find more and more activation of the ductal cells. These cells crosstalk with the fibrogenic cells and with inflammatory cells, leading to inflammation. Couple of words, and I will conclude with this, on extrahepatic signals. This is because the liver is the center of a network that includes the adipose tissue, the gut, and also the muscles. So here we have already heard how the adipose tissue is important, and it generates insulin resistance. Adipose tissue generates adipokines. These are a number of different molecules which regulate metabolism, inflammation, and even fibrosis. There are some molecules that have been investigated at a higher extent. Adiponectin is good because it reduces insulin resistance and it downregulates fibrosis. Resistin, it modulates mostly inflammation, and leptin generates inflammation. We had already talked a little bit about the gut-liver axis, and just to point out again that from the gut there is the release of a number of components, including the pathogen-associated molecular patterns that activates TLRs and lead to inflammation and fibrosis. And we are currently obtaining data showing that myostatin, that is a myokine, may actually impact on the liver, especially on the fibrogenic cells, inducing fibrogenesis. And finally, another possibility that we have to explore is to stimulate fibrosis resolution. If we interrupt the injuring stimulus, we have the activation of fibrosis resolution. We already discussed how the macrophages are important, and this occurs through induction of hepatic stellate cell apoptosis, senescence, and even reversion of activation. So these cells go back to their original phenotype. There are a number of ways that are under investigation. One was recently tested. It's the interaction with the lysyl oxidase 2. That is a molecule that cross-links the collagen. Unfortunately, the trials so far have not been successful. But the point I want to make, and this is my final slide, is that the current knowledge and increased knowledge on the mechanisms of fibrogenesis is generating a number of translational efforts that hopefully will lead to more therapy and more targets for therapy of NASH. These are just my collaborators, and with this I thank you for your attention.